The overall goal of this procedure is to generate native chromatin immunoprecipitation sequencing libraries for nucleosome density analysis.
This method can answer questions in epigenetics, such as revealing epigenomic features in a cell population at a single nucleosome level, and resolving heterogeneous chromatin signatures into their constituent elements. The main advantage of this technique is to utilize MNase property of preferential access to open chromatin region to generate a measurement that combines MNase accessibility with histone modification. Generally, individuals new to this method will struggle because they are not able to obtain optimal MNase digestion.
Begin this protocol with cell preparation as described in the text protocol. On day 1, thaw each cell pellet in a 37 degrees Celsius water bath for 10 seconds. Then transfer the cells to ice. To each cell pellet, immediately add an ice-cold 1x lysis buffer plus 1x PIC to a final concentration of 10,000 cells per 20 microliters. Mix 10 times by pipetting up and down without creating bubbles. Working on ice, aliquot 20 microliters per well of the resulting lysates into a 96 well plate. Cover the plate with a plastic seal before incubating on ice for 20 minutes. Label the plate as MNase and record the wells to a template key. Just before 20-minute lysis is complete, dilute the MNase 1 enzyme with MNase 1 dilution buffer to a final concentration of 20 units per microliter and keep it on ice. Working on ice, prepare the MNase 1 digestion master mix.